to share her screen. The next speaker is Anna, I'm probably going to slaughter this, Matynia? How do you pronounce it?
Yeah.
And she's an associate researcher of ophthalmology at the Stein Eye Institute at UCLA in Los Angeles. It is quite early, or you've got one of the better time slots, I guess, now. No, it's quite early there. So she'll be discussing investigating vascular microglia interactions in a mouse model of retinal hemangioblastomas.
Okay, can you see my screen?
Yes.
Okay, great. So first of all, I'd like to thank you for inviting me to speak here. I'm very happy to share the progress that we've made in our model of retinal hemangioblastomas since 2 years ago. This is my disclosure slide. I do have a grant from the VHL, and I'm very happy for that so we can continue our work, and a separate grant not on VHL. So I'm going to talk to you today about retinal manifestations that are important in VHL. So the retinal hemangioblastomas that occur in patients are often the first symptom that leads somebody to go to a doctor and diagnosis of VHL if they don't know that they have a family history of it. The hemangioblastomas occur both in the brain, retina, and spinal cord, and we're kind of considering these to be equivalent types of pathologies, although we are aware that they may differ based on their site. So to tell you a little bit about, let me get a pointer on here, laser pointer, and whoops, it didn't. Okay, so I'm sorry, I want to just change that color because you won't be able to see it otherwise. Okay, so in patients, retinal hemangioblastomas are not lethal, but they can lead to vision loss through retinal hemorrhage, exudation, and scarring. They do occur at about the same frequency as renal cell carcinomas, and so they are a major problem for patients with VHL. Here's a fundus image of a human eye. This is the optic nerve, and the hemangioblastoma is down here. So this is quite a large one. You can also see that there's a lot of dilation of the vessels that lead to feed this tumor. The tumors are highly vascular. You can see this on a fluorescence angiography, the optic nerve head here, and the hemangioblastoma, which is just filled with blood through this mechanism to visualize blood vessels. Hemangioblastomas are typically large and highly vascular, and they're often located in the superficial retina layers and project into the vitreous. And this is what can cause a problem because you can get some shearing between the vitreous and the retina, causing hemorrhage. And of course, you can have exudation because you have angiogenesis in the tumor. So years ago, very much years ago, there was a lot of work done on patient biopsies of tumors from the retina and from the brain. And what they showed in these patient biopsies was that it was very vascular, so you can see a lot of blood vessels here, but there were also a lot of stromal areas with what they described as giant foamy cells of unknown origin. Other studies, and they're listed down here, showed that VEGF, CXCR4, HIF, and ubiquitin were expressed, indicating that there's angiogenesis, hypoxia, and immune cells in these biopsies. When they further dissected these biopsies, they were able to show that the tumor had a loss of VHL, so a loss of heterozygosity. The patient's own mutation was first copy loss, but the excised tumors themselves had a loss of the second allele. And when they dissected these out, they showed that there was intact copies and intact copy of VHL in the vasculature, but the so-called good allele had been lost in the stromal tissue. So this goes with what Tim Hsu also said, is that the vasculature changes throughout the body may not be the primary target, but may be a secondary effect. So in these retinal hemangioblastomas and cerebral hemangioblastomas, it seems like the vasculature still maintains an intact VHL copy, but the stromal tissue, which is as yet of unidentified origin, does not. So our interest in using a mouse model to study this is to try to understand what the original cell type is that leads to tumor formation. And these are the tissue is being characterized as having giant foamy or stromal cells. And we know that one allele is inherited, the other is acquired, and that only certain cells and tissues acquired this second kit. So it suggests that there's the genetic loss, but there's also perhaps a specific niche for hemangioblastomas to form, and that this niche needs cells that have a capacity for cell division and dedifferentiation or redifferentiation. So hemangioblastomas don't really occur in the skin, although we've heard that there are some changes in the vasculature. They occur in the CNS. So it suggests that there's actually something very specific about retinal and brain tissue, maybe secreted factors or other cell types that are specialized to those areas and not throughout the body. So keeping that in mind, we're interested in looking at endothelial cells. I'll often call these vascular endothelial cells because we have other endothelial cells in the eye, and microglia, the resident immune cell of the retina, because these are phagocytic cells that might become giant bony cells. But we're keeping in mind that there are other cell types in the retina, and we do need to think about this. Okay, so we know that if you delete VHL in mice, that it's embryonically. So people have made conditional knockouts of VHL mice that are loxed with the LOX-P site. And these mice are completely normal as far as we can tell or have looked at. But what they allow you to do is to introduce a pre-recombinase and it will excise exon 1. So this is the two LOX-P sites flanking exon 1, and this is the post-excision. So one of the questions that one has to ask is, what are the cell types that one should target? So we have our candidates that we're interested in, but people have tried candidate knockouts before. We wanted to let nature take its course and biology take its course. So we used what we termed an agnostic approach. So we injected pre-recombinase delivered through viral expression systems into the vitreous. We used a specific serotype of AAV2 that has general tropism to the retina, to retinal cells, and we used a general promoter that allows expression regardless of what cell type. And then we used EGFP to also visualize where it was being expressed in early stages. So we were able to inject this virus and see that it did express shortly after injection. And so we figured out the viral dose that we needed to deliver to get expression of pre-recombinase and hopefully recombination. This is 100-fold less virus. We then also checked our retinas from these mice, not our own, but from the mice, to see if they had excision of VHL specifically. So we have a PCR that we can do for exon 1, and this should occur, we should see this in every single retina that we look at, because our pre-recombinase should excise only in some cell types and not in every single cell, just because of the number of cells and the expression localization. So we can see that in all of our retinas, we have exon 1, so we have cells with intact VHL. But then we can see in our retinas that received pre-recombinase, we see this excised transcript, but not in retinas that did not receive pre-recombinase. So we know that we are excising VHL from some cells, although we don't know which cells at this point in time. That's a future study. So then we know that we had these mice that we could look at for retinal hemangioblastomas, and the first question obviously was, do they get hemangioblastomas or a phenotype similar to that? So we did in vivo imaging to look for blood vessels, hemorrhage, discolorations, and tumors, and also fluorescein angiography to look for leaky vasculature and abnormal vessel formation. And we were able to see such things. So this is a normal mouse fluorescence angiography showing the optic nerve head, the large blood vessels that come out, and then all of the other blood vessels that are underneath it. We were indeed able to see things like either small leaks or possible anastomoses and large leaks. But when we looked through our cohort of animals, some of which were wild-type mice, so they should not be models for VHL with Cree expression, or the floxed mice, which are our model, we can look at the phenotypes we were able to see. So we looked at discolorations, hemorrhage. We didn't really see a difference between our controls and our experimental animals. We got a discolorized lesion in our controls, but not in our is increased vascular lesions in our VHL floxed, excised mouse model, but not in our controls. This was, but it is indicative. So we moved forward to look at the actual histology of the retinas. We looked in retinal vertical sections and whole mounts, where you get a better idea of the entire vasculature. And we looked at both of these for blood vessels, IB4 that stains endothelial cells, and microglia, the resident immune cell. So this is an example of abnormal vasculature that I showed 2 years ago, where what you're looking at is the retina in cross-section. These are your photoreceptors. These are some internuclear cells, amacrine and bipolar cells for the aficionados. And out here is the retinal ganglion cell layer. These are the cells whose axons form your optic nerve and go to the brain. This layer has what is termed the superficial layer of vasculature. There's another layer in here, and an intermediate layer here, and a deep layer here. So this is a control mouse with the photoreceptors, intermediate, and ganglion cell layer. You can see stained in red blood vessels. So this is what you would normally expect to see in the amount of vasculature, and in green, microglial cells. So on a vertical section, this is what you would expect to see. And in our VHL floxed animal model, you can see that there's all sorts of abnormal going on. It's a huge blood vessel. It's multiple layers of cells. Microglia are surrounding it. They're more vasculature that's going deeper into it. So this is highly, highly abnormal and nothing that you want to see in your eye. This differential interference contrast shows that there are red blood cells inside the lumen of this vessel, so it's a functioning blood vessel. So there's only a limited amount of information that you can really see. Think about, I'm going to use a visual aid, an eye. The retina is on the inside, so we're looking at the vasculature. We can take out the front part of the eye and then make cuts in the retina and then lay it flat, because you can't really look at a globe. So we can make these cuts, form these petals, and then look at the retina. So this is stained for blood vessels. You can see that there's large blood vessels that go out to the periphery and a lot of smaller vessels. This happens to be a control eye, and on the other side over here is a mouse model for VHL showing the hypervascularization that occurs in almost a quadrant of the retina. So this is a huge hypervascularization or hemangioblastoma. We can analyze these, and just to orient you, we can analyze these excluding the optic nerve head here. We can look at the central, mid, and peripheral regions, and then also whereabouts in the eye this occurs and where the hypervascularization occurs. So analyzing these samples from a number of animals. Here I'm just showing you the concentric peripheral, mid, and central parts of the retina, the optic nerve head, and the petals, representation of the petals, and the localization in the animal. The legend here is green. The vasculature looked normal. Red, we had hypervascularization, vascular growth, or hemangioblastomas. And in the blue were ones that we felt we couldn't call just by visually examining them. So we quantitated the volume filled by the vasculature of the retina in the peripheral sections, because that's where most of our experimental animals, as opposed to the control animals that are the asterisks, most of our experimental animals had these growths or hemangioblastomas in the peripheral section, most likely due to where the infections are occurring. So we quantitated the amount of vasculature in control animals, and you can see that it fills up maybe 10 to 15% of the retinal volume, slice volume. And if we look at the experimental animals, for ones with hemangioblastomas, there's hyperproliferation. So I'm going to show you what these look like in a look. So this is shown in red is the blood vessels, and in green are the microglia. So we want to know what's happening with the microglia. So you can see very nice, clear sparse blood vessels, and you can see these microglia have a cell body, but then they send out all of these processes. So this is called a ramified microglia, and it's considered to be actively surveilling, but not actively engaged in repair or damage control. And you can see that they also do sit on top of the blood vessels, so they're watching out all over the retina. So this is a sample of normal tissue, and this is an example of what we felt was an incipient hemangioblastoma, early formation of additional vasculature that has grown on top of the superficial layer and looks like it's starting to protrude into the vitreous in these two samples. And you can see compared to the last sample, it's very abnormal to have this sort of tangled knot of blood vessels. And the other thing you can see is that in parts of it, we have normal microglia, but around these blood vessels that are so abnormal, we have a different shape and perhaps higher number of microglia. So that was an early stage of hemangioblastoma formation. And again, like that vertical section, this is nowhere near normal, and it's nothing that you'd want to see in your own eye, but we were glad to see it in our mice eyes. So this is out at the periphery where you typically don't have these larger vessels. And you can see all of this angiogenesis. It looks like a bowl of spaghetti in here. And then the other thing you can notice is that you're really not seeing those ramified microglia. These are all activated or more active, engaged in some sort of alteration to the environment to try to keep normal function in physiology. So we were able to, so this is very preliminary results. This is just like from last week. So we're quantitating the number of microglia to see if we get recruitment to the area or if we just get an activation. So in our control samples, we have about the same number of total microglia as in our affected samples. When we look at the number of ramified microglia, it's also very similar. But if we look at the amount of these amoeboid or activated looking microglia, you can see that it goes up a lot in our affected samples. So we're doing further analyses to try to quantify this a little better and to look at the boundaries, the borders of where the abnormal vascular growth occurs versus where the normal vasculature appears, to see if we see transitions in the microglia and get a hint as to what may be happening. So, basically, I've shown you in our mouse model that we have abnormal vascular growth and what happens to the microglia. I think I hear a click. Sorry, so I'm almost done. So our next steps are to try to determine whether it's vascular endothelial cells that overcome microglial surveillance, or whether microglia themselves are transformed and allow or direct unregulated angiogenesis, or whether they both contribute to this abnormal hemangioblastoma growth. People can ask me about that more later. So just in summary, we used an agnostic approach. We found functional changes that were indicative of experiment, but not of hemangioblastomas, but we see this massive vascular growth and a microglial alteration. So we're hoping to use this mouse model to identify how these two interact to cause the hemangioblastomas. And with that, there's my acknowledgement slide, and I will thank Steve Nuisnowitz and the VHL Alliance. And I'm going to exit out of this so I can see you.
Okay, thank you so much, Anna. We don't have any questions at this time in the questions and answers thing, but I have questions, of course, since that seems to be my function here. I do have a question, so I thought it was really interesting. Do you think that the mast cells are the cause or the effect?
Which cells?
The mast cells.
You mean the microglia?
No, sorry, the microglia. Sorry, I'm spacing out. Microglia, duh.
Right. So our hypothesis is that the microglia are potentially the cells that are transformed and cause it. They can direct angiogenesis, and that's what they do during development. Mm-hmm. The other thing is they are phagocytotic, so they can become those giant foamy cells, but they clearly would have to undergo some sort of transformation. I think there's data out there showing that the foamy cells, stromal cells, are of mixed origin. So we think they have the capability of doing that. And we also think that similar to other cancers, they could be important for preventing uncontrolled growth. Other thing that makes us point or think about microglia is that these are the immune cell in the CNS.
Right.
So that could potentially explain why hemangioblastomas grow in the CNS, is because it's the microglia that reside there and become these cells. And maybe in peripheral tissues like the kidney or testes or adrenal glands, that other cell types take on do this.
Mm-hmm. Fascinating.
But
Fascinating.
are aimed at dissecting that out.
Have you quantified, because obviously you have more endothelial tissue, have you quantified the number of microglia versus the, I don't know, surface area? I don't know what you would measure exactly, the volume of the endothelial tissue, because you see more, but there's also more
Correct.
endothelial tissue. So
right.
What's that relative ratio like?
So a couple of things. So first of all, we don't know if it's interacting with the endothelial cells or pericytes. So that's something that we need to look at. We will do that analysis, but it's a much more sophisticated level analysis. So I would say at this point in time, if we just did a straight density of microglia per vascular volume, that would possibly even be lower because we have so much vascular volume. But what we're going to do, and this is, we're talking with some people in Australia that have put out a really beautiful paper on this, is to use a different software so that we can actually quantitate the interaction surfaces between the two. So not necessarily the number of microglia changing, but the amount of physical interaction between microglia and a blood vessel. So that's stuff that we have started, but are not yet really at the point of doing that analysis, but it's underway.
Okay, wonderful. A question just to come in. I think it's a general question. It's talking about the immune system in general. Can you draw that connection to the microglia? The individual who's asking it is asking, do you think that there's an entire immune dysregulation that's occurring here, or is it specific to the microglia?
Well, okay, so two levels of answer. First of all, since we inject only into the retina, we assume that it's just going to be localized to the retina. So it's limited in that sense. However, the fact that we have angiogenesis means that we're going to have leaky blood vessels, and we can certainly have peripheral immune systems coming in. So I anticipate that we will find other markers of non-CNS
origin
function.
Yeah, interesting. Now, Eric Jonash had another question, and I think this is also a fascinating topic, is are you eventually planning on using the AAV, the adenovirus delivery of VHL, to impact the phenotype on your model?
So that would be a rescue experiment that's being suggested. And yes, but I think we want to know a little bit more first. So for example, one of the next series of animals that we're going to inject, we're going to do fluorescence activated cell sorting of retinal cells, isolate out the endothelial cells from microglia and other cells, and try to find out which cell population has excised VHL. It could be one, two, or multiple populations. If it's microglia, then what we're going to perhaps look at doing is transplanting in healthy microglia with VHL to see if that rescues.
That would be fascinating. Wonderful.
So possibly be a cDNA genetic expression, but also cell type directed studies.
Okay, well, thank you very much, Anna. Fantastic talk. Really nice.